Hello and welcome. We are Emre and Karam. Are you a researcher struggling to find meaningful patterns in your complex genetic data? Do you want to discover disease biomarkers without needing to write any code? In this video, we're excited to introduce BiomarkX, a web-based tool designed to make powerful biomarker analysis accessible to everyone. Let's dive in and see how it works. When you navigate to our website, you can either sign in to save your analysis history or simply continue as a guest. For this video, we'll log in with a sample user account. Once you're in, you'll see this main interface. You have two options to get started. You can upload your own file with the Browse button, or you can use our built-in demo data. For this tutorial, we'll use the demo dataset for Alzheimer's disease to explore all of the tool's features. Let me click on that. Now, in step 2, we can see the information for our demo file. Now, in step 3, we need to select two key columns from our data. First, the column that specifies the status of our samples, like patient or healthy. For our demo data, this is the diagnosis column. Next, we select the column containing the unique ID for each sample. Once these are selected, you have the option to normalize your dataset before moving forward. Let's make sure you clicked the Normalize button. First, we choose a pipeline. For this video, we'll select the MogaNet-style pipeline. While you can adjust various parameters here, we'll keep the defaults and click Normalize. Once processing is complete, step 4 opens up. In step 4, we can see a bar chart showing the distribution of the different patient groups in our dataset. BiomarkX allows you to select two or more groups for multi-class comparisons. However, for this tutorial, we'll focus on comparing two groups: Alzheimer's disease patients against the healthy controls. AD represents the Alzheimer's patients and control represents the healthy individuals. We'll select both AD and control from the list and then click Analyze. After this, you'll see an optional step to handle class imbalance. You can choose to skip this step or click Configure Resampling to refine your data. Let's click Configure Resampling. Here, you can select resampling methods like SMOTE or ADASYN and tweak their parameters. For our setup, we'll choose SMOTE and click Apply Resampling. Now for the exciting part. In step 5, we choose our analysis type. BiomarkX offers a wide array of methods categorized into statistical tests, classification analysis, dimensionality reduction and visualization, and survival analysis. For our first example under classification analysis, let me select logistic regression to run a SHAP analysis, a powerful machine learning method for understanding feature importance. Once selected, you'll see the default parameter settings. We will keep the default parameters for now. In step 6, we can exclude any columns that aren't needed for the analysis. With everything set up, let's click Run Analysis. The analysis may take a few moments to run in the background. Once it's complete, you'll receive a notification. Let's click on the notification to jump straight to the results. Great! Here are the SHAP analysis results, giving us a list of important biomarkers from a machine learning perspective. What if we want to compare this with a traditional statistical method? Let's use the Perform Another Analysis button to quickly run a second test. This time, we'll select ANOVA. We'll keep the same settings and run the analysis. Perfect. Now we also have the results from ANOVA, which ranks biomarkers based on statistical significance. So now we have two different biomarker lists, one from SHAP and one from ANOVA. How do we decide which ones are the most reliable overall? This is where BiomarkX's Combine feature becomes incredibly useful. Let's click the Combine Biomarker List button. We'll tell it to combine the results for our AD and control groups. The resulting table shows us a consolidated ranking. It takes the results from both the SHAP and ANOVA analyses we just ran and creates a final consensus-based list. This helps us focus on the most promising targets for our research with much higher confidence. What's more, BiomarkX offers integrated KEGG and GO enrichment tools right on this page. You can choose between KEGG pathway analysis or 3 different GO enrichment options: biological process, cellular component, and molecular function. Let's click on Perform KEGG Pathway Analysis. As you can see, the platform immediately processes our biomarker list and displays a detailed pathway table, highlighting significant pathways along with their overlap counts, p-values, and associated genes. You can also download these results directly as a CSV file. Right after this, we can also validate our biomarkers against trusted external databases. By clicking Validate Biomarkers externally, BiomarkX fetches cross-references from resources like OpenTargets, Jensen Lab Diseases, and EWAS Atlas. This gives us instant validation on disease associations and evidence scores for our key targets. Finally, clicking Generate Analysis Report downloads a comprehensive PDF summarizing our entire workflow, from consolidated biomarker lists to all individual plots. You can also track and manage your work via the user profile. Under Analysis Results, you'll find all your saved analyses, where you can view details, download reports, organize them into custom lists, or continue your work anytime. Let's click View Details on our latest run. Here, the pipeline breaks down the execution details into clear Information and Results tabs. Just like on the primary output page, you can also use the Continue on this analysis button to extend your workflow anytime. As shown on screen, all your visual plots, metrics, and continuation options are readily accessible whenever you log back in. So to recap, in just a few minutes, we went from raw genetic data to discovering significant biomarkers, creating a consensus ranking, and generating a comprehensive PDF report, all saved automatically to our user profile. To analyze your own data and explore these features, we invite you to visit our website. Trying BiomarkX is completely free. Thank you for watching.